continuing to the course Basic Biology for Engineers and Big Data Analysis. Uh, we are just taking some small baby steps to learn the underlying basic biology concepts and also how they are relevant and going to be applied for the investigations in the omics technologies. So I'm just taking you to some very basic concepts, but those are very essential, especially when we are talking about the field of genomics and proteomics. We are talking about studying all the genes of a given system or all the underlying concept comes back from the original genetics. And I think that's where it becomes very important for us to think about how to clarify all our fundamental concepts. So I'm going to talk to you about some basic concepts of Mendelian genetics. Some of the genetics rule which depends on the probability rules. How this information lies into the chromosomes, what is the basis of inheritance, how some of the chromosome, if they're not having normal shape or structure or the compositions, how that could result into the abnormalities, how DNA replication happens. So some of these basic concept, but as I go along and give you some examples, you will see that they have some rationale and some logic about how the genetic concepts and the genes, how they're going to get transmitted from one to other generation. All right, so let me just start first with the basic concepts of the genetics. And okay, so the first point which I want to talk about some of the basic terminology and concepts of genetics, and then with some examples it will be clear to you that why we are discussing this context. So what are the heritable traits, like which properties, which traits are getting transmitted from one to next generation? So many times things like, you see the eye color from the parents to the child, or the hair color. Some of these things are the morphology or the phenotypic characteristics, which we see already, but there are large number of genetic traits which are heritable from parents to the offsprings, right? And which are the principles which are going to allow that these factors or these traits which we are talking are going to be transmitted. So to understand these kind of possibilities, Gregor John Mendel, he studied the rules of heredity, and based on some very simple and elegant experiments, he made some observations which are still underlying and the foundation of the field of genetics. So some of these characteristics, if I just highlight here, different type of eye colors, if you see, from the brown, blue, green, or gray, or looking at even the hair color or the type of, the hair, sometimes it's curly, sometimes it's, straight. Many of these are what we see for the same phenomenon. These are heritable variations. So we see variability in our eyes, in our hair, and likewise many characteristics which are having variability. So in little bit in biological technical terms, when we look at the morphology or these characteristics which we can visually see that, like these, the eye color or the hair color, those are phenotypes which we see appearance. But what is the genetic makeup of those? That comes under the genotype, and that's what is studied under genotype, which is an organism's full hereditary information or the gene level information. So just refreshing back what we spoke yesterday about the cell. And as we talked, the human body is made of billions of cells. Every cell which is shown here is having this small nucleus and this plasma membrane. In between, we have all those organelles which are important, which we studied yesterday. Now, this nucleus, which is the central part, which you can see now it's enlarged here, is having all these chromosomes. And these chromosomes, now if you enlarge here further, their compositions, these are forming the stretch of the specific sequences. Those are known as genes. So what is genes? Genes are the sequence of nucleotides at a specific locus. Just give me a minute to allow some participants. Yeah, so the genes are the sequence of nucleotides at a specific place or the locus where, on the chromosomes they are located, like in this case here. And Mendel thought that those are heritable factors, some factors which can be passed on from the parents to the offsprings. Even for the same genes, there could be different alleles which are showing the variation. Like the same gene which is going to be inherited, but we are going to take from both the parents. So the eye color, for example, the same gene is going to be inherited from both the parents, and those for that gene, those are alleles which are harbored, and those are going to show as a two copies of that gene. So there are different theories of hereditary, like how genes are going to be transferred from the parents to the offspring. One is blending hypothesis, which is, are the genetic material from the two parents are going to just blend the way you see the paint colors, like the blue and yellow paint, and whether that makes it the green color. Or particulate hypothesis, which got more attention, which leads to the ideas of having a genes. For example, if you look at the deck of cards, and the from the player 1, player 2, player 3, you're passing those cards, and likewise from the parents to the offspring to the grandchildren, you're passing those genes, and these are getting shuffled in the process because of the meiosis which is happening at the chromosome level, and this information is getting passed from one to next generation. So Mendel, he was, not a scientist by profession, but he was studying these garden peas out of his curiosity for looking at, from one to next generation, what kind of behavior they have. So he was actually a monk in 1843, and he was more interested in physics and mathematics. And after studying in Vienna, he came back and started working in this monastery where he was growing the pea plant in his garden, and he chose pea plant because pea plant has various characteristics. Like some were looking at white colored pea plants, some were purple colored. Their heights were different. Their pot shapes were different. So they have, he observed they have different characteristics, and why they are different. Sort of curiosity, he started thinking about some of those experiments in the garden peas, and his observation and especially his mathematical skills of thinking about doing these experiments actually led him to follow from one generation to the next generation how the plants, when he does the crossing, and then those seeds, when he grows and sees the the phenotypes of those flowers in the next generation, or the overall pea plant in the next generation, what changes are being observed. And he was keeping a good tracking and the note of that, and as a result, when he observed many generations and many traits, he was able to follow some patterns. And those patterns he then underlined as the law of inheritance. So one thing which he did very smart, it was he chose those pea plants which is having only one characteristic at a time. So he chose those as a true breeding variety. And what does that mean? That if you are taking a flower with one color, uh, so that flower, when you take male and female plants, and then you are doing the cross, uh, only you're going to see that one characteristic which is going to be observed in the next generation. So based on these experiments, he was trying to determine that what is the units or the factors which are passing from the parents to the children or the offsprings. And these units or factors at that time, he did not knew about them, but now we know these are the genes. And so an individual inherits one such unit from each parent. So again, these are alleles now, which is for a given gene, you have two alleles coming from each parent, and those are going to be inherited and being shown in the next generation. So his first law, which is the law of segregation, that he looked at two different variations of the pea plant. And now let's not worry too much about the plant or the colors, but think about a little bit more mathematical notations here. If this was a dominant characteristic, and this is a recessive characteristic, like the purple flower color and the white flower color, and if you choose the notation as a capital P for the purple, And because it is dominant, you are showing capital P, but because the recessive trait is white, you can now show the small letters, small p, but you can definitely choose any other notation like capital T, small t, or capital R, or any other letter you can choose. So now these gametes are coming from these purple flower or the white flower, which is capital P or small p. So when he took these flowers from the parent generation or the P generation, he did this cross. And after doing the cross in the first generation or the filial generation F1, he was able to observe only purple flowers. So all the flower which came from this hybridization or the crossing, all of those showed only purple flowers. So this was, quite surprising that, okay, like I'm taking two different colors, but in the first generation, all the flowers are only showing one trait, and that's why the purple color became dominant, and he termed those the dominant characteristics. And so now, if he took again these purple flower from the first generation, F1 generation, and did further cross, and now I'm sure you would have played the checkerboard and this kind of matrix. So now if these gametes, which is capital P and small p, which is composition of this purple flower, if you take the male, the sperm from the male plant, and the eggs from the female part of the plant. So now you take these gametes, capital P, capital P, and do this cross yourself. What you'll observe that the capital P characteristics is present in at least three flowers, and recessive characteristic or small p is only one flower. So this part, when he observed now the second generation, that ratio of the purple flower versus white flower is 3:1. So now he realized that they're segregating. So initially there was some dominance of some trait, which was in the first generation, but in second generation they started segregating. And that's where then his law of segregation came, that two alleles, which is allele capital P and small p, for the heritable traits, they separate from each other during the gamete formation and the subsequent generation. And these are now, we know, these are alternative versions of a gene, which we now term as the alleles. So just to clarify the terminologies again, and I'm sure all of you from engineering background, this might be getting slightly overwhelming to you, but let's just think more logically here. So we are looking at these segregating population of these flowers. A simple cross, when you can decipher the gametes, you can just take some notation for those. And possibilities here are capital P, capital P, small p. So now you see this 3:1 ratio. Whenever you have these purple flowers versus white flower, this is what you are saying as a phenotypic ratio. So in the second generation or the F2 generation, he was able to find the ratio of the flowers appearance wise 3:1. But if you think about the genetic wise, the genetic composition wise, which is known as genotype, so only one of them, which is this one, is having homozygous dominant trait, which is having capital P. Whereas this one, the white flower, is only recessive characteristic, also homozygous. Homozygous means, like, the both the alleles are either capital P or small p. Heterozygous means you have one capital P and one small p. So that is the heterozygous. So the purple flowers are actually having one homozygous and two heterozygous. So that's where the genotypic ratio, if you see, those are 1:2:1. There are some more complex genetic concepts, which is known as back cross and test cross, which can determine what could be the genetic composition of the gamete composition of that, which I'm not talking right now. But I'm just giving you the flavor that by just observing simply the phenotype of the plants from one generation to the second generation to the third generation, Mendel was just writing down those numbers of the how many flowers are coming from which type of traits, and based on these, then he did the similar experiment with the tall pea plant versus dwarf tea plant, and he observed the same trait, same patterns of the segregation. And likewise, he looked at 7 characteristics of the pea plants, and all of those were showing very similar kind of behavior. And but so over the years, then scientists have been investigating, and Mendel's rule, it still holds very true. And now we know that the units or the traits which Mendel was talking, those are the genes, and the traits, which is capital P, small p, which is coming, that is the alleles. So now if we think about in that way, so these are the chromosomes on which the alleles for a purple flower or allele for the white flower is present. And depending on how we know in the next generation, the gametes are going to harbor these copies, either going to be, purple is going to show the dominant behavior, and it can be, further segregate in the subsequent population, the way you have seen in the genetic cross. So dominant allele has the effect, but recessive allele does not have effect, and that's then people further try to investigate what could be more molecular mechanism or basis of this behavior. Why only the purple color what we see is coming because of what, and why that is dominant. So they now looked at more at the nucleotide sequence level, and if you recall yesterday we talked about these base pairs, right, in the nucleotide sequences. So we have ATGC compositions. Now, if you look at the same chromosome, what we are talking for the purple flower versus white flower, it's only one nucleotide, which is, in the nucleotide sequence, only one base pair change, which you see from C to A. So the entire sequence for the gene which is going to give rise to that flower color is actually exactly same for the purple and the white, except that C is replaced, cytosine replaced with adenine here, right? That's the only change. And as a result, this is going to produce some enzyme in the series of reaction, which is going to generate purple pigment or purple color. And that's why you see the purple flower, whereas that sequence is missing here, and as a result it can't do the enzymatic reaction to generate the purple pigment, and that looks like the white flower. So this is now how the science progresses, how the research progresses, how we understand the basic concepts and take them forward and establish more of the molecular layers to that to validate those basic concepts. Mendel further looked at the not only one characteristic, but now he started thinking about looking at two characteristics at a time. So earlier he was only looking at, either the seed is yellow colored or green colored, or seed is round shape or wrinkled shape. But now he is looking at two characteristics at the same time, that is known as dihybrid cross. When we are looking at one characteristic at a time, like purple flower versus white flower, that was monohybrid cross. It means we are looking at only one characteristic. So now in this case, he's looking at a seed which is yellow colored and round shape, or a seed which is green colored and wrinkled shape. So now when you do this kind of cross, and again, I'm sure while you're all from mathematics and engineering background, all of these could be very well derived without worrying too much about the plant notation and coloration. You can just, once you know the gamete notation, then it actually becomes very much like mathematical rules and becomes very similar. And as I go along in the probability, you will then see that how much mathematical basis is involved in doing genetics. And that's why genetics test right now, which is all in the clinics, they're all are based on very good rationale, and they all work. So now in this case, you are looking at the gametes from this particular seed, which is going to generate capital Y, capital R, or from the green and wrinkled shape, which is recessive in trait, small y, small r. So in the first generation, F1 generation, you're still going to see dominance of only these yellow and round seeds, which is going to generate this gamete shape here. So Mendel was not sure that now what he's going to see in the second generation, because in the monohybrid, when he was looking at only one characteristic, only one was appearing as a dominant. So purple versus white or green versus yellow, only one is coming as a dominant. Now he's looking at two characteristics. It is green versus yellow and also round versus wrinkled. So what is going to happen in the second generation? So he just thought mathematically there could be two possibilities, that these characteristics are depending on each other. So in the second generation, I'm going to see that they are dependent, or these traits may be independent, like they are not depending on each other, and the seed shape, versus the seed color, both are independent traits, and they're going to assort independently. So after so the possibilities, what he had in his mind, if they're going to have the dependent characteristics, then the ratio will remain the same, even if it is a dihybrid cross. But if they are independent, they are going to segregate separately, like two separate monohybrids, then it will be characteristic will be very different, and we might get a ratio of 9:3 to 3:1, unlike 3:1 if it is dependent. So based on his, he already made predictions, but when he saw these results, he could see that, so he was counting number of seeds which came from those cross, And he could see that these ratios of these seeds came 9:3:3:1, which proved that hypothesis of independent assortment is correct. These characteristics, which is the seed shape, which is looking at wrinkled shape or the round shape, or their coloration, which is the green versus yellow, they are independent and they are going to assort independently. So each pair of these alleles are going to segregate independently of other pair of alleles during the gamete formation, and that set up the basis for his second law of independent segregation. Let me now show you this nice video clip from this link, which will refresh you all the concepts.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase y, and the recessive green allele as a lowercase y. The uppercase y always overpowers his lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
All right, so I hope now you are refreshed, and just to summarize of this part one. So Mendel formulated first his law of segregation, where the two alleles for a given trait, they're going to separate from each other in the gamete formation in the subsequent generations. In the law of inheritance, he stated that there are two or more genes which are going to assort independently, and he showed with those crosses and showed the ratios will be 9:3:3:1. So I hope so far it is, while it was little biology, but at least now you see the rationale of doing these experiments and how they have, a lot of rational basis, and which is actually, the concepts which remains same irrespective of whether Mendel tried to do those experiments long back, or even now people are trying to establish the molecular basis of these experiments. So now let's take a couple of other examples, which is more familiar to us. So far, we just talked about the pea plant, and I hope this is now clear to you that we are talking about single gene, one gene, and there are two alleles of that gene, which is showing some variation. And while even if there is a one capital P present, then that is going to show the purple flower. But if both small p's are there, then it will show the recessive characteristic, which is the white flower. Now, similar thing also happens in our blood grouping, right? Now, I'm sure you will feel more interested that what we are talking is actually having the basis to our own genes and our own various properties which we talk. So the blood grouping, as you know, the ABO blood grouping is quite complex, but you'll be surprised it is just derived from one single gene. It is having multiple alleles, and those are very important. Now, if you just think about the blood group AB type, so in this case, A and B, these are two different alleles, but those are capital A, capital B, which is heterozygous in nature, right? What we are going to now talk about, so that term known as codominance, it means both A and B is having equal dominance. Uh, in case of only blood group A or blood group B, it will be only dominance of A allele or B allele. Or when both of these alleles are not dominant, it will be small I, which will be blood group O. So we will not elaborate more further on the multiple alleles, which I'm sure will relate all of you that how these genetic rules which we are talking are applicable to us in our day-to-day life. So now when we talk about ABO blood grouping, as I mentioned that, any individuals, which all of you are aware, can have, one of the four type of blood groups, either blood group A, blood group B, blood group AB, or blood group O. Now, when we are using these terms, these are actually, when we talk about red blood cells on the RBCs, there are two sugar moieties. It could be either carbohydrate A or carbohydrate B. So that's what we are going to term them as a type A blood. Or if the sugar type or carbohydrate type B is present, then that is known as B type blood group. If both of these are present, like A and B carbohydrates on the RBCs, that is AB type. And if both are absent, then that is known as O type. So let's see like how we can denote these notations now. And these are one gene exists in more than two allelic forms. So now we are talking about multiple alleles. What we talked about in Mendel's pea plant case was one gene, two alleles. What we are talking about now, one of the human gene or the human blood grouping property, one gene, multiple alleles. So here now notation is based on this carbohydrate or the sugar moiety, which is present on the RBC. So now if I is the notation for the dominant characteristic of this, where capital A and capital B is showing the carbohydrates which is present on the RBCs, and if they are absent, that is none. And this is shown with a small I. So now phenotype, let's talk about first, which we are all aware of, the blood group A type, B type, AB, or O type. Now, how they look in terms of appearance, this is RBC. On that, if this was the carbohydrate A, this is how the appearance looks like. If another type of carbohydrate is present, which is B type, it will look like this. Or if both of them are present, it will look like AB type. Both of them are absent, it is O type. Now, genotypically, how you can represent that? Because you are going to show a dominant trait coming from this carbohydrate, which is A. So either it will be I capital A, or it can be I capital A and a small I, right? Still, it will show the same dominant trait as you have seen already in the heterozygous cases. Or it can be I capital B, or I capital B and a small I. In case of AB, it will be both of them will be heterozygote, will be dominant, and I capital A, I capital B, or it can be recessive, small. So how we denote these the blood grouping, the genotypes, and how people do the testing in the labs. I'm also going to show you. In fact, in some of the engineering undergrad courses, when we teach here, we also give them, to do this test, in a controlled and tested environment, where they can also check their blood groups and they can appreciate themselves that how genetics actually works, even in the, without having much big laboratory setups. So if you think about now, the parent alleles could be A type, B type, or O type. One parent, and second parent may have allele A, B, or O type. And accordingly, now these offspring will have any of these possibilities of these blood groupings, right? So let's see this video to see experimentally how you can, determine what type of blood grouping it may have. And then I will give you some more rationale about how to decide those blood grouping.
An adult human has about 4 to 6 liters of blood circulating in the body. Blood consists of several types of cells, including white blood corpuscles or WBC and red blood corpuscles or RBC floating around in fluid called plasma. There are certain protein molecules called antigens located on the surface of the red blood cells and antibodies which are in the blood plasma. The differences in human blood are due to the presence or absence of these antigens and antibodies. Individuals have different types and combinations of these molecules. According to the ABO blood grouping system discovered by Karl Landster, there are 4 types of blood groups: blood group A, blood group B, blood group AB, and blood group O. Rh or rhesus factor is found on the RBC surface in most people. Like A and B, this is also an antigen, and those who have these antigen are called Rh positive. Those who lack this antigen on the surface of RBCs are called Rh negative. To conduct the experiment, we require 3 monoclonal antibodies, anti-A, anti-B, and monoclonal D, kept in an icebox. Lancets, spirit, cotton balls, toothpick, and glass slide. Procedure: swab the finger with cotton balls dipped in a spirit and take blood sample using a lancet as shown. Take 3 separate drops of blood on a glass slide. Place a cotton ball on the finger to stop the blood flow. Pour 1 drop each of anti-A, anti-B, and monoclonal D on each drop of blood respectively. Mix the monoclonal antibodies with the blood sample properly using a toothpick. Make sure you take a new toothpick for mixing each drop. When RBCs carrying one or both antigens are exposed to the corresponding antibodies, they agglutinate, that is, they clump together. For example, in this slide, agglutination occurred on the first and the third drops, which corresponds to antigen A and Rh factor. So the blood group is A positive. Thus, we can identify the blood group by noticing the agglutination on the corresponding drops.
So I'm sure you have, at least you are familiar with the initial part when you go for your blood grouping test, and you see that how they're testing, but then you do not know that how they are determining and telling the what blood group you have, right? So let's take this situation of AB blood group. So what we are talking about, these enzymes or carbohydrate moiety A present, or B present, or AB present, right? So antibodies have been raised against these antigens. So they are only going to recognize either only A antigen or B antigen, or you have raised antibody for AB. So now when you are going to look at where antigen and antibody reactions will happen, so when these reactions will happen, then they are going to show agglutination, as you can see these precipitate kind of things which is happening. So let's start with maybe A first. So when you have added antibody A and that is reacting with antigen A, so in this case now you see agglutination happening only in this case here, whereas no agglutination happening with the antibody B. So this is only determining that the person is having blood group A. In other case, when you are not seeing any reaction happening with anti-A antibody, but you see a reaction happening with anti-B antibody, then you can say this is blood group B. When both of them are reacting, like antibody B is also showing the reaction agglutination pattern, antibody A is also showing, then it is AB. When both of them are not reacting, that is O blood group. Now, in addition to these, there is one more factor, which is rhesus factor, which comes with the antibody D testing. So now, if it shows reaction, that is known as positive. If it does not show agglutination, that is known as negative. So in this case now, anti-D antibody is used. So now you can say this is O positive or O negative. I hope things are very simple and clear to you. Just to summarize, we're talking about how the genetics rules are applicable to our real life as well. And blood grouping is one of the very interesting example, which we all go for our blood test. And even with simple test, which is based on the antigen antibody, what we are looking at for a given gene, there are multiple alleles present, which could be based on the carbohydrate present on the RBC, which is A or B or both A and B, or absence of both of them. Absence is going to determine the O type blood group. Both present is giving rise to AB blood group. Only A type is giving blood group A. Only B type is giving blood group B. On top of that, we're also looking at an additional factor, which is rhesus factor, which is being used with tested with antibody D. And if that is showing positive pattern, agglutination, or negative, accordingly we say O positive or O negative. So these are some interesting and basic example, but likewise we have many examples when you will see the genetics rules are one could follow in our day-to-day life. And most of them have actually a good mathematical basis, and they are based on the rules of probability. That's why you can use some of these genetics rule and make prediction that what may happen if these are the parents, and if they have going to have a children, what possibilities of inheriting some of these traits, which could be good trait or bad traits in the subsequent generations, right? And those could be looked into based on the probability rules. So I don't have to tell this audience, which is already from coming from engineering, that how you measure probability between 0 and 1. And that's of course is, it's like, you are doing a toss and you have two possibilities. And likewise, same thing goes when you are looking at these gametes from the sperm and egg. And we were talking about those gamete characteristic of the dominant or the recessive traits. So let's say that is coming half and half, capital R or small r, and same coming from the egg and same coming from the sperm. And accordingly, if you look at now the what Mendel did the cross in the similar way with the probability basis, now you got 1/4 of the homozygous dominant, 1/4 of the homozygous recessive, 1/4, and 1/4 here means half of the heterozygous, which is going to show the same pattern in the phenotype, which will come from all of these three, which will show the same phenotype. This will be different. So that is, not only applicable to just looking at monohybrid crosses or simple cross, what we just showed, but also is applicable even for the dihybrid cross, like what was more complex crosses when we are looking at multiple characteristics and how they are going to assort and looked at independently. So in this case, to simplify these kind of questions and these kind of problems, one could consider that rather than thinking about dihybrid cross, can we think about two monohybrid cross separately? So in this case, as you recall, we were talking about the seed color and seed shape. So if you just dissect out and just you think about, we are talking about one cross, which is only looking at seed color. Seed color is yellow versus green. And now for that, these are my, the gametes, which I'm going to do the Punnett square cross. And they're going to show me that, one is forming capital YY, small YY, and then the 50% of the heterozygotes. These are my gametes coming from this monohybrid cross of seed color. And likewise, my cross of seed shape is going to give me these gametes. So now if you want to look at probability of a specific type of trait coming in the subsequent generation, so I asked some of these questions to our students here, like, what could be probability of F2 genotype with this particular composition? So for doing that, you don't have to think about all the colors and the entire cross, but simply look at the probability of this particular gamete, which is 1/4 here, probability of this gamete, which is also 1/4 here, and then you can say this is only 1/16 probability of this gamete to occur. Likewise, capital Y, small y, capital R, if you look at this particular gamete, which is heterozygous here, half probability here, and capital R is 1/4 probability. So that gives rise to 1/8. So you can actually look at the complex cross, which was looking very biological, is actually very mathematical. You can simply look at now probabilities of these gametes to occur, and you can derive and tell yourself that what are the kind of seeds which are going to happen in the F2 generation, and their possibilities of the coloration and the shape, right? So this is how the genetics and the rules of probability are very important. There are a lot of genetic tests which is following these kind of basic principles. Now, while Mendel did his experiment, Mendel was still not very popular. He made all those observations, but there are many scientists who were actually trying to test his observations after several years and decades. And then actually they confirmed that what Mendel did actually is holding true even on different organism, different model system. And that's how Mendel got much more recognition later on after his lifetime. And now he's considered as the father of genetics. But one more scientist, he contributed immensely, and he was following Mendel's experiment. His name is Morgan, and he was trying to replicate what Mendel did, but in a different type of experimental model. He was looking at now what can be done in the Drosophila or the fruit fly. So when you eat banana, and if you leave the banana peel and you see those small flies, those are actually fruit flies. And if you enlarge their eyes, they look very pretty. They are red in color. So people do the experiments on Drosophila fruit fly because you can generate these in very short time. You can do the crosses very fast, and they come with different traits as well, like the pea plants. So Morgan was working on these Drosophila. Just to tell you, there are a lot of scientists, they do research in Drosophila, and there are good stocks which are maintained. Even IT Kanpur one faculty, Dr. Sinha, he does a lot of work on the Drosophila genetics. So there are good cultures available for doing this. But long back, when Morgan did the experiment, he was looking at how to replicate now Mendel's experiment. Mendel did on the plant, but now can I do something on the insects and the animal system? So now he was looking at these flies, fruit flies, female and male, and the female was having the red eye, and he chose the male which is having the white eye. So now think about the dominant and recessive characteristics which we talked in case of purple versus white in the Mendel's P plan, and start thinking similar kind of situation here. So now in the first generation, he observed all the offsprings, whether it was male or female, every fruit fly contains only red eyes. In the F2 generation, he observed that now three of the offspring out of four have the red eyes and one is having white eyes. And among the three, two of them were female and one of them was male. So it's not only that we are looking at dominant and recessive characteristic. There's also something which is where these traits for this gene is located. Is this located on the X chromosome or Y chromosome? Even that plays an important role. So he not only replicated Mendel's experiment and he proved that Mendel's rules are actually correct, but also he made observation that these genes are located on the chromosomes on either X chromosome or Y chromosome, and they are actually the heritable factors, which is also determined from which chromosome they are coming from. And similar thing actually also happens in human. If you think about chromosomal basis of sex in human, so we have 46 chromosome, 23 pairs, and 44 plus XY chromosome in the male and 44 plus XX in the female. Now, the gametes which will be formed in the female will be all having X chromosome only, whereas in the sperm we have either X chromosome or Y chromosome. So after the fertilization, the crossing, it will be either XX or XY, right? So the male will be XY, female is XX. So whether a boy will be born or a girl will be born will be depending on the sperm characteristic and sperm's way of fusion with the egg, and that determines what is going to be the fate in the next generation. So sex of an offspring depends on whether the sperm cell contains X chromosome or Y chromosome, and this is also then the traits which are located on X chromosome are going to pass, the traits which are located on the Y chromosomes are going to pass. So now we have, with the subsequent years of research, we have good understanding that on each of these chromosomes, like X chromosome and Y chromosome, there are many genes which are only exclusively located only either on X chromosome or Y chromosome. And there are at least 78 genes which are only on the Y chromosome. And there are quite large number of genes, which is almost 1100 genes, which is on the X chromosome. So these genes, they are carrying different characteristics. And some of those are also, very important in our practical life when we see day-to-day life. Some people are having red-green color blindness, or some people show the Duchenne muscular dystrophy, or night blindness, or hemophilia. Many of these characteristics, these are something which is linked to different, the genes which are coming on different chromosomes. So I'm sure all of you can read this particular number that it is 15 number in the green color in the red background. But these are the kind of tests which people also do when they go for railway exams and other kind of military exams. They also want to make sure that individual is not having these color blindness, right? So these diseases or these traits are actually linked to a specific chromosome, and these are known as sex-linked traits. Now let's talk a little bit something which I'm sure you can use with more rational logic. If you have a pen and paper, start doing this yourself, and you will feel much more comfortable that genetics you can now learn, and you have a rationale about knowing how these things work. So what we are looking at now, a practical situation, a male and female when they are marrying, and what kind of probability of the children they will have who can carry this particular trait for the color blindness. So we are only looking at one disease, red-green color blindness, which is X-linked disorder. It means this particular disease happens for a given gene defect, which is happening only on the X linked to the X chromosome. So if there is a mutation happening, which is on the X chromosome, that is, we are denoting for the simplicity's sake on the as a small n recessive allele. If it is normal dominant allele, that is shown as the capital N. So here there are three possibilities that the female was normal and male was colorblind. So at the time of marriage, female was normal, male is colorblind. Or female is a carrier. You can't see that she is carrying this trait, but she is actually carrying this problem, but not apparent, not dominant, and male is normal. Or third possibility could be female is carrier of color blindness and male is actually colorblind, right? So these could be possibilities. And this is actually a point to also highlight and discuss that while in India still we have such a luxurious weddings, so much money is spent on various type of expenditure during the wedding, but still no genetic test happens as a mandatory part. Kundlis do match, but genetic tests don't happen. I for one of my collaborative work in Tunisia, a small country which probably many of you have not heard about, but of course they play good footballs. If you are football, if you like sports, you know them. Uh, so there, I went for a collaborative research work, and I was giving a talk, and one of the scientists was hosting me. Uh, there, one key scientist was supposed to come and meet me, but she did not come. So I was wondering what happened, because I went all the way from here to have meeting with their groups, and this person, why she's missing. So they told that she's going to get married, and mandatory they have to do genetic test, and she only got that date test, unfortunately. So then she has to go for that test. Otherwise, they'll not allow for marriage to happen. And she apologized, but she said she has no choice than to be there for that test today, and then she can meet me next day. So think about the awareness, the level of awareness what other countries people have, and how important those are. Before getting married, they want to know about these details rather than only just matching the kundali and thinking about luxurious weddings, right? So now if you already have brought the sheets and pens with you, you can think about this cross yourself, right? So let's say this female is normal on both the chromosomes. The trait which we are talking is dominant, capital N, and it is showing normal characteristic. In the male, which is XY chromosome, so female is denoted as XX, male is shown as a XY. In male, we have this recessive characteristic, which is a small n. So now if you take the Punnett square to look at the probability of this children or the offspring to carry these traits, so the gametes coming from the sperm will be X small n, like from this one, and the capital Y. The gametes coming from the X will be X capital N, X capital N. So now if you do Punnett square, you can yourself see that now the only female is going to carry this particular recessive trait. So they will not show, because they are not going to show the dominant characteristic, because until unless on the both X chromosome it was a small n, then only the female will be colorblind. So in this kind of case of wedding, now in the first generation, nobody will know that any of the children are carrying the disease or not, because they will all look normal, and the female or the daughters are only carrying the trait, recessive trait. Now, another possibility could be that male looks normal, so as female looks normal. And after the wedding, when they have offspring, one of the boys, he starts showing the colorblind pattern. So it was not actually, thought of, because both of them were normal. But now if you look at the molecular basis to that, you can yourself determine that 50% of the male are going to carry this trait, and 50% of female or the daughters will be carrier. So none of the girls will show in that first generation the problem, but the 50% boys are having probability that they will carry this particular colorblind trait. Now, third possibility could be that the female is carrier and male is colorblind, and now after the wedding, then 50% of the boys will be having the color blindness, 50% of the girls, the daughters, they will also have the color blindness, and within the female population, the 50% will be the carrier. So now you see, like you can make this prediction yourself. Now you are engineer, but you can do a lot of things which the doctors are going to do, right? And genetic tests are going to tell. These are all very simple logic, simple rationale, and now you can think about the distribution of XY chromosomes and certain genes which are linked to those particular chromosomes. And if we know about their behavior, those genes, we can actually look at the trajectory of those genes and find out what are the possibilities of the offsprings to carry those disorders. Additionally, a similar concept is also applicable for hemophilia, which you might have heard about the royal family story in UK. And I'm not going to talk in detail about that, but just again, similar phenomenon is also observed in the hemophilia, which is a disease based on the absence of one or more of the protein which is required for the blood clotting. So whenever we get an accident and have the blood is coming out, so for blood to clot, there are some proteins and factors are required, and if those are absent, then you will see a lot of blood loss. This particular phenomenon is actually linked to also X-linked recessive disorder, like what you just saw in the color blindness. And if that problem is there, then individuals are going to take a lot of time to show the clotting, and that could have a lot of bleeding, excessive bleeding. And that was a phenomenon observed in the royal family in UK, and that's how a lot of scientists started working on it, and they try to decode what is happening from one to next generation, and is there any kind of chromosomal basis for these disorders. So many of these disorders, now we know that they have genetic basis, they are linked to some genes, and these genes are located on some chromosomes. So there is many possibility which also happens within the chromosomes of for certain genes, that some genes can be deleted, some genes might be moving and getting inverted in the process, some gets duplicated, like the same BC gene now becomes 2 copy here, or in some cases it is exchanging one part here to there, and one part from here to there. So as a result, that is getting translocated. So during the process of meiosis and other genetic recombination events happening, there is possibilities of alterations of chromosome structures. And sometimes we also see, like certain chromosomes, we are talking about 23 pair of chromosomes. Some chromosomes also show more than 2 copies, like in this case known as the Down syndrome disease. The 21st chromosome has 3 copies, and therefore we see this child which is not normal. Edwards syndrome is also trisomy, or the 3 copies of chromosome 18. Then in Turner syndrome, we have absence of one of the or both of the copies of the X chromosome. In Patau's disease, it is a trisomy of 13th chromosome. It means 3 copies of the 13th chromosome. So these are the practical situation which we do see these children, we do see these individuals. And if the genetic test could have happened, probably this could have been easily decoded that what problem they have. Let me show you this clip to you, and let's then talk about this.
Hi.
Hi.
Your son?
Yeah.
What? क्या हुआ इसको?
It's a chromosome glitch. हर इंसान में जेनेटिक कोड होता है. क्रोमोसोम A, C, G, T को मिलाकर हर शरीर में 3 बिलियन ऐसे कोड्स होते हैं. कभी-कभार जेनेटिक म्यूटेशन की वजह से इन 3 बिलियन में से किसी एक कोड में T की जगह अगर गलती से C ले लेता है, तो उसे औरो कहते हैं. लकी बॉय, हाँ?
So I'm sure how nicely Vidya Balan actually explained this, probably everybody can understand that. This is a disease known as progeria, or Hutchinson-Gilford-progeria syndrome. And now we know there is a gene which is the lamin A gene. If there's a defect in that, then we see this kind of characters which is oro, right? So of course, there are ways to defend that, but still, if you think about these kind of children, one could actually predict them early and try to avoid these problems. But now we know there are scientific basis to that. There are some gene defects which result into this kind of baby. So yesterday I talked to you about the composition of DNA. So I'm not going to talk in detail about the DNA. You already know that we have these 4 base pairs, which is 4 bases ATGC, which is forming hydrogen bonds, 2 hydrogen bond with A and T, 3 hydrogen bond with G and C. We also have the sugar, and then we have the phosphate. They are forming the phosphodiester bonds, and these are 2 anti-parallel strands, which is dictating the DNA double helix structure. This rule of having 2 hydrogen bond with A with the T basis, or G with the C basis, having 3 different hydrogen bond, that is comes under the Chargaff's rule. So reason I just refresh you on this, that we also talked one concept yesterday, that in the cell cycle, there is the mitotic phase, and then we have an interphase. During the interphase, the cell is trying to prepare for the division to happen. And that time you have multiple phases like G1, S, and G2 phase. So S phase is where the DNA replication is happening. It means DNA is trying to make its more copy. So when DNA is trying to make more copy or its own replicate, what are the possibilities of that replication to happen? That was another scientific observation people had, and many people proposed different hypotheses. Different scientists had different hypotheses that if the parent DNA looks like this dark blue colored, so a possibility could be that one of the after the first replication cycle, when you want to make more copies of the DNA, this dark blue gets separated, and now another light blue is getting synthesized because the cell is continuously providing those material and nucleotides for it to make the second strand. So now it makes double helix, which is having one dark blue and one light blue, and likewise the second DNA looks same. In the next round of replication, now you will have one is keeping this particular dark blue and the light blue, but the light blue is also synthesizing another light blue. So now you can see this kind of pattern where two molecules looks like like the first replicate, and two new looks like, which is the light blue in color. So this and likewise you can look at the dispersive model, which is not following this pattern, which is they are all randomly dispersed, or conservative model where you can have one DNA which is still looking same, the dark blue one in all the subsequent replicates, but the other molecules are appearing. These are the possibilities. Given the short time I have, we will just take briefly one experiment which proved that the DNA replication happens in this manner, which is known as semi-conservative replication. So what they did, they took bacteria, grew in the culture, which was labeled with the 15 nitrogen heavy isotope, and then they transferred that to the 14 nitrogen lighter isotope media. After centrifugation is done, they are looking at the density pattern of these DNA, which is now labeled with N15 and N14. And what they observed, that light band should appear here, heavy band should appear here, and hybrid band should appear in the middle. So they had only light band, which is like this one, or the hybrid band, which is having this kind of composition. That's what is observed in the density centrifugation experiment, which proves the Michelson and Stahl experiment that semi-conservative model of the DNA is the best explaining this phenomenon. Of course, we can talk in more detail, but let me just wrap up this right now. So what we studied so far started some very basic biological concept of genetics, of theory of inheritance, but then we also looked at how logical it is to have these genes transmit one characteristic from one to next generation. A diploid individual, they contain two copies of these genes, and each parent is transmitting that one copy or the allele for that one gene, which is going to be transferred to the kids for the next generation. A trait may show up or may not show up, like we have seen in the color blindness. In the first generation, we may not see those characteristics, but it can still get passed on to the next generation. And now we may see in the second generation that the childs are hemophilic or colorblind. Also, we have looked at the how different type of possibilities of mutations happening, or the physical and chemical disturbance which can damage these chromosomes. This process happens quite a bit in the meiosis process, and then these can also generate into the chromosomal abnormality, and different type of syndromes are there based on these chromosomal abnormality. So I hope you despite your varied background, not coming from biology, but at least you got some flavor that how genetics and genes and alleles and the chromosome test, genetic test, which we all talk, those big things come from these simple experiments and very elegant experiments and very basic biological concepts. Please feel free to refer to this book, Campbell Biology book, where most of these figures have been taken, some of the video clip I showed you, and of course some of the images. So I hope you have got some flavor of biology, and this is going to give you more foundation for us to discuss further how these genes could now be studied at the genome-wide scale using more bigger technology, omics technologies, and next generation sequencing, microarrays. These are all the fancy technology which we are using right now, which is generating big data, omics data set. But the basis of that comes from these, the basic scientific foundation, which is from long time has been established. Thank you very much.